this video, we'll talk about some basics in microbiology to get you introduced to this subject. So, what is microbiology? Microbiology is the study of microscopic organisms, such as bacteria, viruses, fungi, and protozoa. Generally, we divide the organisms, these bacteria, viruses, and fungi, according to their characteristics. So in bacteria, we use gram stain, either positive or negative, to categorize them. In viruses, we depend on their genome, DNA or RNA, whether it's single-stranded or double-stranded, to categorize them. In fungi, they're either divided into molds, which is multicellular, or yeasts, which are unicellular. We also have the hierarchy of classification. We used to put each organism, including us humans, into its category. We took it in high school, if you remember. So from highest to lowest, it's domain, kingdom, phylum, class, order, family, genus, and species. You can use this mnemonic to help you remember it. Dina's kids prefer candy over fried green spinach. Spinach is pretty underrated, by the way. So if we start from the top, we have 3 domains on this planet: bacteria, archaea, and eukarya. Both bacteria and archaea are considered as prokaryotes, whereas eukarya are considered as eukaryotes. The main differences between prokaryotes and eukaryotes are absence of cytoplasmic structures such as mitochondria, Golgi apparatus, and endoplasmic reticulum in prokaryotes. And then we have absence of cell wall in eukaryotes, except for fungi and plants. Also, they differ in the structure of the ribosomes. After that, we go to the kingdom. Both bacteria and archaea have only one single kingdom, which are bacteria and archaea, respectively. And then we have the eukarya, and it has 4 kingdoms: protists, fungi, plants, and animals. And then we continue the classification like this, like we mentioned before: phylum, class, order, family, genus, and then species. For now, we will focus on bacteria. How do we name them, basic structures, and gram staining. Any bacteria's name is composed of 2 words. The first one refers to the name of the genus, and the second one refers to the species name. We'll take Staphylococcus aureus as an example. So Staphylococcus is the genus, and aureus is the species. Also, the name of the microorganism might refer to its shape, like in our example. Staphylo means a bunch of grapes, or a cluster, and coccus means round cells. Aureus means it looks like gold in color when we culture it. Other than cocci, the round cells, we have rods or bacilli. And then we have spirochetes, which are curved. And then we have diplococci, which is basically 2 cocci, or basically 2 round cells. And then we have coccobacilli, which is a mixture of both a rod and a round cell. And then we have vibrio, which is a curved rod. Then we have streptococci, which are cocci in chains. And then we have the staphylococci, like we mentioned in our example, which are large cocci in irregular clusters. And then we have tetrads, which are cocci in a packet of 4, from the name tetrads, tetra, which means 4. Now we're going to discuss the bacterial architecture. Since bacteria are considered as cells, that means they have genetic material, a cell membrane, and a cytoplasm. In addition to that, they have a cell wall, a flagella, which is used for locomotion, and then we have pili, which is used for attachment.